presentation, which is about chronic ischemia-reperfusion injury in Long COVID and M.E. CFS. And without further ado, let's hear from Doug.
So thanks very much indeed, Chris, and everyone organising, and my apologies for not being there, and also for the almost complete absence of any genetics in this presentation. So as you'll be aware, partly from the title, we've been working away at this problem of anomalous clotting, and by we, I'm pressing my next slide button, nothing's happening okay that way. We, I mean my long-time collaborator, Respiratory, and I have been working together for approximately 11 years, research from Stellenbosch. And so it's very much in trouble. We've been working on this problem of anomalous clotting for quite some time. And I'll take you through a little bit of the backstory. And what I'm going to talk about is the symptoms of long COVID and M.E. CFS, which of course you know are manifold, and despite the fact that it seems implausible, it could be a unitary explanation for many, if not all of them, we think that there actually is, and that therefore begins to give an explanation. We think it starts with the formation of these unusually resistant to fibrinolysis microclots, which block up capillaries. And recently we recognized that some of the sequelae of that probably reflect a chronic as opposed to an acute, which is well known, ischemia-reperfusion injury. And that, of course, leads to some possibilities for treatment. So if we look at the vast list of symptoms, this is long COVID, but as everyone knows, M.E. CFS shares the great majority of them. Of course, not all are happening in every person. Some are much more prevalent than others, in particular fatigue, post-exertional malaise, as used to be called, now referred to as post-exertional symptom exacerbation, and the recognition that blood disorders and clotting are actually coagulopathies really quite at the heart of these things, and that low blood oxygen is a consequence of that. And that it is, of course, a multi-system disorder. And you can list all of the observables and start referring to some of them as causes and some of them as effects, because they do have a temporal relationship. And what we're going to focus on particularly today are the coagulopathies, a little bit about platelet hyperactivation. It's bound up with endotheliitis, and then some of these effects, particularly fatigue and PDSE. And of course, recognizing that inflammation is at the core of much of this, and that many of these things that appear as effects then feed back to become causes. And that causes essentially a vicious cycle in which A causes B and B causes A, and this leads to an amplification of symptoms that is that we actually are the cause of pretty well every kind of disease. And the benefit of those who aren't into clotting, and apologies for those who are, the sort of 101 version of this is that there is a large clotting cascade with two main pathways referred to as extrinsic and intrinsic, leading to thrombin, which is molecule 2A in the pale blue, which is a proteolytic enzyme that acts on fibrinogen, the second most abundant protein in plasma, after albumin, and we'll look at fibrinogen in a second, that leads to its polymerization and the clot that we see, that eventually the clot can be broken down by plasmin, and the D-dimer, which we'll mention a few times, is a result, but actually, as it turns out, not really an important cause of anything we're interested in here. So if you look at the fibrinogen molecule, which is about 45 by 5 nanometers, a cigar-shaped thing, what thrombin is going to do is to remove serially these two little peptides, which in here on the A subunit as in red and blue, and the consequence of the removal of these two little peptides is that there's enough free energy to cause the thing to self-associate and make much bigger fibers, so that instead of being 5 5 nanometers in diameter, spaghetti-like clots, you'll see some pictures of shortly, are going to be typically in the diameter of 50 to 100 nanometers and much longer, many microns or tens of microns. So we're going to have our fibrinogen here, and it's going to join up to make fibrin, and then ultimately be degraded by fibrinolysis. And all of that's well established in the textbooks and happens, and the normal fibrinogen is of alpha helices. What we discovered some years ago, and indeed further years ago than this, but I'll start this story here in 2016, is that the normal clotting, if you just take some ordinary plasma, add thrombin to it, or indeed purified fibrinogen, you get these clots that I've referred to before, looking a bit like a plate of spaghetti. Whereas if, in this case here, you add a minuscule amount of bacterial lipopolysaccharide, you get these two disgusting looking messes here that we used to refer to at the time as dense matter deposits. And you can see that what happens in these cases is that there's a kind of bifurcation. Some of the fibers get smaller, some of them get much larger, and clearly very different from the normal clotting. And yet this amount of LPS is 1 molecule per 100 million fibrinogens. And you might think these guys are off their heads, what they want, homeopathy or something. And the answer is no, there's no thermodynamic problem whatsoever. From a thermodynamic point of view, it's just like prions. Here is the normal form of the prion protein, PRPC, that I'm acquainted with. And what is the actually significantly more stable, indeed, resistant to proteinase K, version with no change in primary sequence, the amino acid sequence, PRPSC. This has a much increased content of beta sheets, which is why it's resistant to fibrinolysis, because it's really a completely different shape, and it stains importantly with a molecule thioflavin T that is normally not fluorescent, or it is quenched, but when it's bound to the amyloid structures of the PRPSC, the prion protein, it lights up like bilio in the bright green stain. We'll see some examples, not of prions, but of the sort of staining we're talking about here. And so from that paper, when we look at the control, there's not really much staining with thioflavin T, it's visible, but when you start adding, sorry, it's come off here, LPS in the top right-hand corner, and you see massive amounts of staining. This is adding LPS binding protein, which of itself doesn't do too much. And when you add it together with the LPS, you see that it removes the ability of the LPS to do this thing. So it's very clear that this tiny amount of an exogenous substance, in this case from an infectious agent, is capable of causing this massively amount of anomalous microclotting. And as I say, they're much more resistant to fibrinolysis. We'll meet later that they resist tryptophanization. And because people get a bit obsessed about D-dimer, I've put this in here, that the amount of D-dimer you get depends significantly upon whether you go to normal fibrin, which can be degraded fairly easily, or whether you go to the anomalous fibrinoloid fibrin, as we call it, because people get a bit worried about amyloids, so we've tried to change that name, which is only slowly going to D-dimer. So whether you have a lot of D-dimer or don't have a lot of D-dimer, it doesn't tell you anything whatsoever about the fact that you might have a lot of this anomalous fibrinoloid fibrin, which is resistant to fibrinolysis because it makes some more root. So if we're making these anomalous amyloid type fibrin microclots from fibrinogen in the way we've seen, and I'll show you in the next slide, that we've shown that the spike protein of SARS-CoV-2 alone, purified recombinant no virus present, can induce this also at tiny concentrations. The failure to get rid of them by fibrinolysis is going to lead to the fact that they can go and block up the microcapillaries, we'll see that in a second, leading to a bunch of phenomena such as low tissue oxygen and thrombosis and so forth. Whereas if you can get rid of the clots, which are normally happening all the time, so from the normal clots, not the amyloid ones, then all is well and you don't get non-COVID. And with Risha, we worked out a scale, a semi-quantitative scale of how bad your microclots are, and it's in this paper here. And I'll just draw attention to the fact that when you look at the bright field microscopy, you see this, and you can see that all of these clots that are here are actually stainable with the thioflavin T. So there are no, for practical purposes, non-amyloid clots, because the normal fibrinolysis got rid of them. It's in the case that there are these amyloid clots that the fibrinolysis cannot get rid of them. And so the clots you see in the bright field are actually all the fibrinoid microclots. And as I mentioned before, you can take normal plasma, platelet-poor plasma, PPP, add the thioflavin T, and in this case, at least sometime early on in the pandemic, when not many people have either had COVID or been vaccinated, there's almost nothing in these. Whereas when you add the spike protein, you can see much more formation of the amyloid microclots, and you can exacerbate that by adding some extra thrombin exogenously to the normal plasma. And you can see that some of these clots are really quite big. And then we moved on to look at long COVID, where it became obvious and long COVID, but here's an example pre-COVID, and here's someone who got long COVID, and you can see, I'm not going to go through all of these. There's just a lot of these amyloid microclots that stain with thioflavin T in people who've got long COVID, and the number of people with long COVID who don't stain is significantly above the controls, is 0, right? Everyone who's got long COVID does this. We're beginning to find now that people are getting vaccines and so forth, that some of the people are beginning to stain, or who indeed have had COVID, because of course they've got spike protein sloshing around in their system too. But of the people who have long COVID, we have not found anyone who doesn't have microclots. And then we moved on to ME/CFS, and there's going to be 3 slides on this. This is the reference, and find everything. I'll put on the end where you can find all of the papers. They're all open access. And obviously the question arose when we started working with Amy on this one, who I've done for many years. Question arose as to whether or not, given the similarity of many of the symptoms and the basic mechanistic argument that the microclots can cause hypoxia and fatigue, should they not be present also in the case of ME/CFS? And the answer is yes. Here's the healthy participants. Here are people with ME/CFS, and there's a lot more when you just look down by eye, and when we start actually doing the same with thrombin, you see even more. So there's a lot more stuff lurking that thrombin can pull out, rather than just the natural thrombin that's in the plasma. And above the top is without thrombin, the below is with. So clearly healthier way down here, and individuals with ME/CFS are way up here. And on average, this is the next slide, is it? No, it's not. On average, the area taken up by the microclots in the case of people with ME/CFS is 10-fold greater than that of the healthy individuals, where it's basically next to nothing, apart from this person here, who clearly is not perhaps such a healthy individual after all, but they were presented as healthy. And because some of the vaccines either contain spike or the ability to make it, we wanted to make sure that the vaccine wasn't a confounder. That would have been embarrassing if all the people had been vaxxed here, and all we were seeing was that. But we can see that, at least in this sample, vaccine is not a confounder of the ability to form microclots in our platelet-poor assay, which simply involves taking the plasma, platelet-poor plasma, staining without or with some added thrombin with thioflavin T, and looking at the area taken up by the fluorescent pixels. So it's a very easy assay to do. And so again, apologies for people whose knowledge of anatomy is considerably better than mine, but what happens in the system is that blood is oxygenated in the lungs, goes through the arteries, and comes back through the veins, and in between, in the tissues, the exchange is done with this much greater surface area of capillaries, which happen to be very similar in size to the microclots. So clearly, if you've got a microclot that won't break down and it's got a certain size, simple filtration will ensure it gets stuck in the place that just perfectly fits its diameter. And we've seen there's quite a range in size of these microclots. So if you accept that all of this stuff is going on, the microclots are there, they're trying to get into capillaries, and they're stopping red blood cells from going round and round the capillaries because they've been blocked up by the microclots. Consequence is obviously ischemia by definition, and the consequence of ischemia is hypoxia, and fairly obviously you're not going to be able to make ATP aerobically and regenerate energy. And if you're in a muscular tissue, you're clearly going to get fatigued and because you're just not going to be able to produce ATP at the required rate. And because it's hypoxic, depending upon how long things are hypoxic, clearly any tissue is likely to be damaged by that hypoxia. Unfortunately, it's hard to get those kinds of measurements because they're deep inside tissues and people aren't very keen on having those kind of measurements done. And we wrote all of that up and more than once. And most recently, which is going to be the second half of this talk, there was a recognition that one of the consequences of this kind of hypoxia and ischemia, which could be relaxing and remitting, just like CFS is, and long COVID, is a kind of chronic ischemia-reperfusion injury. So what I'm going to talk about now is what that is and how it works and what the evidence is for it. So when you're in an ischemic state and oxygen is dropping down, first of all, you're going to see a lot more anaerobic glycolysis or aerobic glycolysis, but you're not going to get rid of the lactate and pyruvate that's produced by glycolysis. And when you reperfuse, by whatever means, or allow the oxygen to come back in, that's the reperfusion, oxygen's going to go up. But the big thing that's going to happen is a vast production of reactive oxygen species. And the nub of that is on the next slide, what happens here. So if you look at the standard mitochondrial respiratory chain, classic textbook version, here's cytochrome oxidase, and it's going to do a 4-electron reduction to take oxygen to water. Always hunky-dory. If, however, you've just had a prolonged period of anoxia or hypoxia, the upper parts of the respiratory chain are in a much more reduced form, or in a reduced form, such that when oxygen is readmitted, it can now react here with complex 3 or cytochrome BC1 with a 2-electron reduction to make peroxide, or things like the level of peroxide, and here in complex 1 and ADH dehydrogenase with a 1-electron reduction to make superoxide. And you may say, well, these are just signaling molecules. It's true, they are just signaling molecules at low concentrations. But at high concentrations, and most importantly, in the presence of unliganded iron, which is almost always sloshing about because cells have died and liberated ferritin, and the ferritin has liberated its iron inside it, the ability of hydrogen peroxide to react with ferrous iron in what's called a Fenton reaction liberates the very nasty and damaging hydroxyl radical. And there's a second reaction called the Haber-Weiss reaction in which superoxide reacts with the Fe3 that's been formed up here to take it back to Fe2. So these two reactions together mean, again, that amplification of very small amounts of iron act catalytically to produce large amounts of hydroxyl radicals or reactive oxygen species, but the hydroxyl radicals are really the bad guys. And this is synonymous with, it's not lead to, it's synonymous with so-called oxidative stress. That causes all kinds of other stuff, the inflammation and many other sequelae that are recognized as part of PESE. And in addition, the reason, so everyone is full of bacteria, not just in our guts, but in our tissues as well. A third of the population is carrying TB, it normally doesn't do anything. Half of the population is carrying Helicobacter, it normally doesn't do anything bad. But of course, if they wake up and start doing things, things can get bad. And the things that mostly cause them not to be replicating in people are, again, an absence of free iron. So anything that liberates iron or any kind of trauma can also liberate or wake up bacteria, which we've seen produce LPS, and there's other papers published for the equivalent in gram-positives, which is lipoteic acid, and or if you wake up dormant viruses that also happen to have surface proteins that create the microclots. So we think that dormant or latent infections are absolutely part of the whole story, and there's quite some evidence for that, but that's not my focus today. In addition, and again, I'm not going to talk much about this, and Reece is particularly keen on the platelet side of things and has done lots of measurements. Platelets are dripping with receptors for all kinds of stuff. And what is discovered in the case of long COVID and M.E. CFS is a long COVID thing. A hyperactivation of the platelets is sustained for P-selectin, platelet marker, and it's pretty obvious that there's a fairly large difference between what you see on the left and what you see on the right. And the platelet hyperactivation, again, causes or drives the clotting through one of the two pathways, and that's part of it too, but also does many other things because the platelets have so many receptors. And I have to say, to me at least, platelet biology has been a bit of a neglected area, so we don't quite know entirely all the other sequelae are. So I'm mostly going to concentrate on the microclots today, but I'm going to mention platelets as well. So again, if all of that is true, there are predictions as to things that you would expect to happen and be measurable, and they are. So you predict that venous oxygen levels are low, relatively hard to measure, but people do it, and they are. You predict that there are more thromboses and aneurysms, things get blocked up and veins bulge and stuff like that. And it's true. COVID toes is a particular manifestation where you can actually see that clearly blood is not going round and round in the way that you hope that it would. And as I mentioned before, if you don't get enough proper oxygen reduction in the mitochondria, you're going to produce lactate, which is true, and you're going to see the products of loss activity. Hydroxyl is so reactive in nanoseconds that it reacts with almost anything else. And it also including nitric oxide. And the consequence of HONO, which is the product of the reaction with nitric oxide, that's a very strong nitrating reagent, and nitrotyrosine is going to be the product of hydroxyl radicals when it hits proteins. 8-hydroxyguanine is going to be related, are going to be the product of when it reacts with nucleic acids, and malondialdehyde is the product of when it reacts with lipids. And so, although you can't easily see, or can't see, the hydroxyl radical, you can detect it by the products it leaves behind. And these things are raised in these kinds of diseases. And one of the things that I think people found hard to understand is, whilst you can understand why you might get fatigue because the oxygen isn't getting there, and so as soon as you start running, you're feeling tired out, and or you might get POTS because your heart is doing what it's supposed to do, which is to sense the low oxygen and try and beat faster to do what it normally would do. What people with PESE get is that they get these troubles a day or two later after the exercise, post-exertional. And according to us, that's explicable in terms of the time it takes the cells, particularly vascular cells, to die as a result of the blast of reactive oxygen species that they get consequent upon the reperfusion after you stop trying to do the exercise. And so we think that provides an explanation for the otherwise slightly odd kinetics of PESE. And then if all of this is true, you would expect various things such as anticoagulants, clot busters, including nutritional clot busters such as nattokinase, serrapeptase, and lumbricase, and maybe bromelain, to have some benefits, and there is anecdotal evidence they do, but still no trials. Also antioxidants, which help mop up the reactive oxygen species, and the HELP apheresis, the H is heparin, which they also, Jaeger and others have been pioneering, which remove physically microclots. And all of these have been shown to be of benefit to especially long COVID people, and in some cases M.E. CFS as well. But where they've been done, they've just been done. Nobody's done a control like a proper RCT. So I'll have to admit that it's all anecdotal, and there's all the usual caveats about placebos and stuff. But this said, a lot of people are getting benefit from these things, and that's at least consistent with the kinds of explanation I'm giving here about how it works, what the microclots are doing, and how you therefore can improve if you can manage to get rid of them. So again, looking both at the microclot side and the platelet side, all of these things are potentially areas which, if you can stop them overreacting or misreacting in the case of the microclots, you can expect to get therapeutic benefit. And the so-called triple treatment, which all the doctors are terrified of giving, because if you don't measure the coagulation properly, there's a theoretical danger of people bleeding, and you don't want to do that, are involved in stopping the coagulation itself, usually up here, not warfarin, but other more modern things such as the Pixavan, stopping the clotting happening, relying on the non-zero, much slower, but non-zero rate of fibrinolysis of the fibrinoid microclots, and various antiplatelet antagonists such as clopidogrel and aspirin. The triple treatment, so-called, is an absolutely standard thing in acute conditions, but and has been shown to work but not in an RCT in the case of people with long COVID. So people who get acute COVID either, of course, recover, or in many cases, in some cases, don't even notice it. The demographics of people who tend to get worse with acute COVID are older males who might be a bit on the chubby side, whereas the demographic of people who tend to get long COVID are tending to be female, younger, and often quite thin and very fit and so forth. And there's some evidence that estrogen is involved. It explains the demographic in which people with long COVID are probably 4 to 1, likely to be women rather than men, although other explanations are possible for different immune systems. And over time, it's recognized that probably there's going to be the production of autoantibodies, not least because if you're clotting into these anomalous forms, you're presenting variants of your own proteins that are not the normal conformation that would be there. And so it's recognized that over the longer periods, autoimmunity may well play a role. And just to go back to a slightly older review where we looked at a lot of chronic inflammatory diseases, obviously predating COVID, there's a recognition that all of these things can lead to, bound up with iron dysregulation, stimulating the production of things like LPS, causing inflammation and releasing inflammatory cytokines. Nobody's going to argue with that. The LPS can do that. And coagulopathies. In this case, I've used the example of preeclampsia, which is something else we've been working on over the years. But by and large, all of these steps have actually been shown, but tended not to be put together in what I would refer to as a systems biology model of the thing of interest, and very broadly similar phenomena are occurring in all of these diseases as summarized in review. So getting towards the end here, we think that long COVID, in particular with the spike protein, whether there is viral persistence or not, the problem is in many cases, the spike protein is sufficient to induce all of these things such as the amyloid microclots, leading to the oxidative stress from ischemia-reperfusion injury. And yes, there are undoubtedly some genetic predispositions which aren't yet well understood and will be the focus of interest for this group. But equally, let's not forget the lifestyle factors, which in many cases are going to be even more important. And best of all is if when people design GWAS type experiments, they can try and hoover up some of these as well to really work out what it is that individuals do or don't do to cause them to get long COVID, when in many cases they have often been fit as a fiddle. So to conclude, I think without any doubt now, the presence of these kinds of microclots in a whole slew of chronic inflammatory diseases, many of which I didn't mention, including Alzheimer's, Parkinson's, rheumatoid, and type 2 diabetes is well established. And in particular, from the perspective of the interests of this group, it's been absolutely shown in both acute and long COVID and now in ME/CFS as well. What I've brought along to the party today in particular is the recognition that an extra consequence of the ischemia is essentially a chronic version of ischemia-reperfusion injury, and I've tried to rehearse, albeit briefly, evidence consistent with that view, which leads to the recognition that there might be some novel, or not novel, but treatment options on which one might choose to focus. And I'll just leave this up there, although I'll be able to send around all the stuff to people. You can find links to all of these things on the website I put up there. You follow, as Chris mentioned, the Twitter and hashtag Team Clots. But these are the two most recent reviews where we rehearse essentially what I talked about today. And yesterday there went up quite a long video interview with Risha, myself, and Mark Walsh, which was inclusive. With that, I thank you. You're very kind attention.
Thank you very much, Doug. Now, we're not going to clap. We're just going to do jazz hands or something like that, just to show that the people are comfortable at home. Fantastic summary of your work going back many years now. And it's been very well received on the Twitter sphere. It is not without controversy. So I hope that we can have a vibrant conversation today. Does anyone have any questions here or online? Miriam.
My question is about size.
You'll have to re-say the question, Chris, because I cannot hear it. Just say it when the questioner is asked it.
Thank you.
Right, okay, I'll try to speak up when you repeat my question. So my question is about the size of the microclots in the long COVID patients compared to the M.E. patients. It seems from the data that you showed today and from looking at the papers that the microclot seems to be bigger in long COVID patients. Okay. I'm not sure this is the case. Can you confirm or not?
Can you confirm whether the size of the microclots in M.E. versus long COVID are any different from one another?
So this is to be the subject, all being well, of future studies where we're going to do it much more quantitatively, where we do exactly analyze the properties of the microclots in the different conditions, including diabetes and other things, from the perspective of size distribution, the ability to trypsin. What I put in, but evidently forgot to put in, the fact that the microclots trap many proteins. There's two proteomics papers just come out now under ours, including alpha-2 antiplasmin and all kinds of cytokines. So the reason you don't see these things raised in these syndromes, and people go to the doctor and all of their things are normal, many of their things are normal, is because they've actually all been trapped inside the microclots. And so when you do your spinning to get your supernatant to do plasma assays, actually you've removed all of the things that were there, but they were trapped inside the microclots. So we're going to be doing a proper study with all that kind of detail to answer the question, and I wouldn't put myself out on a limb by saying that we've done it yet and give you a definitive answer. There are differences for sure, but we're going to do it properly.
Okay, thank you very much. Kenny Bailey.
Thanks very much indeed, Doug. I'm new to this field and really enjoyed the introduction to the evidence for a role for microclots. You said that one of the tests for this was venous oxygen levels in patients with ME/CFS. Could you say a little bit about the results of those experiments?
To the extent that they've been done, and they're non-trivial experiments, because you've got to go way down into the right place, yeah, people are reporting very low oxygen sats and venous sats, with comments from their doctors like, "I'm surprised you're still alive," this sort of thing. So yeah, where people are measuring these things, they are absolutely lower than they would normally be.
So if I may follow up, that's what confused me. I wondered if that was a slip of the tongue when you said it in your talk, but if you have tissues that don't get oxygen out of the blood, the venous oxygen levels are higher. So the hypothesis that clots are blocking capillaries, leading to impaired oxygen delivery to the tissues, means the tissues don't get that oxygen. So the oxygen levels in the veins under that hypothesis would go up.
Depends which veins.
Well, I assume from your answer, you're measuring them in the pulmonary artery.
No, so that's what people used to do, but now they do it in a different vein whose name has gone out of my mind, but is discussed in the second paper that's on the screen, the lower one.
Perhaps a follow-up reference.
Yeah, this is exactly the kind of conversation that we should have. So thank you very much, both of you. Yes, Kell.
I'm wondering about the mechanism for the microclot formation in long-term patients. Do you have a model for that?
Again,
Do you have a model for the long-term formation of microclots?
So one possibility certainly is viral persistence, where the thing just keeps shedding spike, and we've shown the spike is sufficient. It may not be necessary, but it's certainly sufficient to induce the microclot, and tiny amounts of it. Remember, one molecule will do a great amount of microclot formation. There is evidence for viral persistence for sure, and it will definitely differ between different people, both in amount and location. In the kind of inverted commas worst case, it may be that there are bacteria in the gut that are actually serving as hosts for the viral replication and spewing out large amounts of spike protein that can then eventually find its way into the bloodstream and do bad stuff. So the short answer is we need to find out much more in detail, that the spike protein alone is absolutely sufficient to do it, and at low concentrations.
So if we may, let's take some questions from online. Shall I read them out, or do you wish to?
Yeah, if you go back a month, should I bring them over, or have you got them?
Shall we start from the top?
Yes.
Am I looking at Q&A or chat, because I can see them?
Q&A, if you can start with the top questions, and if you could answer questions after your talk, that would also be great.
Yeah, I don't know what Clexane is, so I can't answer that one. If someone tells me what it is, I might be able to.
For some people, reperfusion has not improved their health. Why might this be?
Reperfusion has not improved their health. Well, because it causes reperfusion injury. Yeah, absolutely. That's why it might be. I wouldn't expect reperfusion to improve people's health. I mean, in some cases, so people try this hyperbaric oxygen therapy. In some cases, they say it makes them feel better. In other cases, it doesn't or doesn't do much at all. And of course, it can also, in some cases, lead to increased ROS production. So what is absolutely the case is that everyone is different. And there's papers that have sought to group the symptoms of long COVID, and certainly there are 6 very identifiable clusters in one quite nicely done paper. So the fact that everyone is different is not of itself news. Where the microclots go and cause trouble is where the trouble is going to be found. And where they happen to go depends upon the size of the capillaries and the size of the microclots, none of which we tend to know anything much about. Next, with inflammation of the brain, there's CNS there.
Maybe if you could answer that online, if that's okay, Doug.
I can, if you tell me how. Do I just type answers into the? Oh, I see where it says answer live. Okay, I'll do that after the end of our time here.
Okay.
I will send you a copy of my slides. I'll PDF it to make it a bit smaller.
Excellent.
Could the vaccines have a similar effect? Yeah, sure, they're the same thing. And we know that many people are vaccinated with very similar symptoms.
And then I think you've already said that clinical trials are required to prove or otherwise this hypothesis. So that's
Well,
the,
that is what people are expecting. What we're doing now is in the UK with some collaborators showing what has been shown already in South Africa, but apparently you have to show it in the UK. So when we published in the UK that the microclots are prevalent, we'll be able to get the ethics to get rid of them, because we won't get the ethics to get rid of them if we haven't shown that they're there. With my clinical collaborators, I think we've got some funding lined up. But what we haven't got lined up is the ethics, and we won't get the ethics until we've shown microclots.
good. I'm sorry. There are so many questions that people have in the room, but we've reached the end of our time. Thank you, Doug. Thank you, your collaborators.
People know my email. I'll mute and stop my video and stop sharing, and I will answer the questions in the question and answer thing. Sorry.
So we're going to jazz him and say thank you very much for everything that you're doing. So thank you. And next up is Kenny Bailey. And we're just going to set up here. So it may take a minute or two.
So well, that's broadcasting here. Is it broadcasting on Zoom? I'll just go to full screen.
Yeah. So people online, can you see Kenny Bailey's?
It's this so far.
So
So we need to switch.
So which one are you?
Oh, this is
That one.
that's the
Okay.
wonderful. Thanks.
Do you want to introduce yourself?